So now we kind of review the very last content area of the class. I can't hear for all the applauding. The last organ system we looked at was the muscle system. And we started by comparing and contrasting the three different types of muscle tissue. There's skeletal, which moves the bones and is attached to bones by tendons. Then there's cardiac. Cardiac's found specifically in the heart. And then there's smooth muscle tissue, which is blood vessels and viscera. Each of the three is specialized for its function like anything else in the body. So let's look at how they're different. They're different by location, as just mentioned. You find them in different organs. They differ slightly in their function. Here's the deal. All muscle contracts, regardless of what kind of muscle it is. But because of structural differences and how they're put together in their organs, when they contract, they accomplish different function. Skeletal muscle is all about generating force and moving a bone. You can also generate heat by shivering. Cardiac muscle is all about coordinated contraction within the chambers of the heart to pump blood. And smooth muscle can do peristalsis, it can do movement, it can change pressures. They differ slightly in their neurocontrol as well. Right off the bat you have somatic versus autonomic. But even within the three different muscle tissue types, how they are innervated by their motor neurons can be slightly different. And lastly, because they are specialized, they have different structural components. They are the same in that the cells of all three types are fairly long relative to other cells. They all have a sarcolemma. Sarcolemma is their cell membrane, and their cell membrane can generate and propagate action potentials, so it gets a special name. All three muscle tissue types are similar in that they have sarcoplasm, that's their cytoplasm, and their cytoplasm has structures in it and molecules in it that you won't find in other tissue types. And lastly, they're similar in their prefixes, in that because they're muscle tissue, you'll see myo, mes, and sarco. In fact, you see sarco in these two words right here. Okay, and so those prefixes are used in the words that are structural components of muscle tissue. Now, once upon a time, we actually looked at the same slide in a previous chapter when we looked at tissues. And here's the three different tissue types with a photomicrograph of each. Skeletal cells, remember, are cylindrical. By cylindrical, I mean they are long, cylinder-shaped cells, very long. So on this diagram, you can't even see an individual cell. They're longer than the field of view. Skeletal muscle cells are multinucleate. If you look at this slide, there are way more nuclei than there are cells. At most, there's 1, 2, 3, 4, 5, 6, 7 cells, but you'll see way more nuclei. That's because they start out as several different embryonic cells that have to fuse together to form one big skeletal muscle cell. And after they fuse together, their nuclei stay present. Skeletal muscle tissue is voluntary. Remember, it's under somatic control. And those action potentials begin in the cerebral cortex. Skeletal muscle tissue is striated. That dark pink, light pink striping pattern you see comes about because of the way the contractile proteins are arranged inside. And the organs that skeletal muscles make up is a muscle. Now, we learned more about muscles in the lab. Remember, muscle as an organ is the belly, which is the skeletal muscle tissue, the stuff that contracts, and a tendon or aponeuroses. That's the dense, regular connective tissue stuff that connects the belly to the bone. So a muscle as a word, as an organ, has several different structures in it. Alright, then we have cardiac. Cardiac muscle cells are branched in that they're kind of like letter Y's in their shape. Okay, so we can outline one down here. It starts here, goes down like that. That would be that cell right there. Okay, so they are branched. Because of that, between some of the branching, you'll see a little bit of connective tissue sometimes. So it's not nearly as stacked as the skeletal is. Cardiac cells are still striated. They are uninucleate for the most part, meaning one nucleus. So they are uninucleate. They are involuntary, meaning they're under autonomic control. Those action potentials begin in the hypothalamus and diencephalon, and they have reflex centers in the brainstem. What's unique to them structurally is they have things called intercalated discs. Those are these perpendicular lines you see between cells. It's a combination of desmosomes and gap junctions. It's a connection between cells that allows them to share cytoplasm, and it has a lot of tensile strength and is flexible. Cardiac muscle cells within the heart are arranged in bundles, and within given bundles, those cells need to contract all at the same time in a given coordinated fashion. And the intercalated disc helps them do that. Right, the third group was smooth. Smooth muscle cells have no striations. If you look there, you don't see the dark pink, light pink stripes that you see in cardiac and skeletal. They have myofibrils, but they are arranged differently in the cell. Their shape is kind of long and skinny. They're not cylindrical at all, and that shape is spindle. They're still involuntary, autonomic control. And the organs you find them in are blood vessels and viscera, organs that are tube-like. Functionally, when skeletal muscle tissue contracts, it is a rapid contraction, meaning from the time you stimulate it to when they go through an entire cycle to shorten and then relax to their original length, that's a small amount of time relative to the other two. Because of that, in a given period of time, skeletal muscle cells have the ability to do more contractions. So pretend you're looking at the three tissue types and you're watching them contract over 1 minute. In that 1 minute time frame, because the skeletal tissue cycle's so short, it can do many, many more cycles in that minute. And so it tires out easily. When muscles tire, it's because they run out of ATP and their ion balances are off. Skeletal muscle cells can also do things called graded responses, which we'll talk about later. For now, graded response means they don't behave the same exact way with every stimulus, meaning if you give them a different stimulus, they'll respond differently. Cardiac cells have a very moderate, steady contraction cycle, so they don't tire out very easily. That's good if you think that your heart started beating well before you were ever born, and it will beat 60 to 70 times a minute on average until the day you pass. So you don't want those cells to have to tire out. They behave as groups of cells because of those intercalated discs. Smooth tissue has a very slow contraction cycle. It takes these cells a bit of time to shorten and relax to their original length. Because of their slow cycle, in that same 1 minute period of time, they will have fewer cycles, and so they're also very slow to tire. They can contract as big sheets. So they're kind of the opposite extreme to the skeletal system. And skeletal muscles, you'll see in a moment, you have the ability within a given muscle to contract a few cells. Smooth muscle cells work differently in that they'll contract as entire sheets. They also have a stretch and recoil property to them. If you stretch them, they have an automatic response that they do. If you stretch them, they actually will contract in response to that stretch. Alright, so we're at the beginning. going to focus on skeletal muscle tissue. So one muscle is one organ, and let's look at some other little pieces of information about them. Every skeletal muscle in the body has a nerve that innervates it. That nerve, as we saw in lab, comes down. Same rules apply. It's got an axon, telodendria, axon terminals. And where the axon terminals synapse on that muscle cell, we have a structure called a motor end plate. The motor end plate is the part of the synapse that belongs to the muscle cell. And the cell membrane there is highly folded up. That sarcolemma is highly folded up to increase surface area. Otherwise, it's a synapse. Just so the same rules apply. You're dealing with neurotransmitter and vesicles and clefts and all that. Skeletal muscle cells also have an artery and at least one vein. That's for continuous blood supply. And skeletal muscles have insertions and origins. Remember, the insertion is the bone or the structure that moves. The origin is the bone or the structure that does not move. When we look at a skeletal muscle's anatomy, we can see that it's thousands upon thousands upon thousands of muscle cells. And they're put together in a way that was similar to nerves. If you take one muscle as an organ, you can see it's attached to the bone by its tendon, and then this part would be the belly. On the outermost surface of the belly, you have epimysium, a layer of connective tissue around the entire muscle. Slice through the muscle and you'll see that the cells are arranged in bundles. Around each bundle is a layer of connective tissue called perimysium, and the bundle itself is called a fascicle. Go inside a fascicle, pull out one muscle fiber cell, and around an individual cell you'll see endomysium. Now remember, skeletal muscle cells are very, very long. They're relatively thin. They're multinucleate. They have sarcoplasm. Sarcoplasm is their cell cytoplasm is a little different from others in that they have specialized molecules. This is the biggie. Myoglobin is a molecule for muscle cells that allows them to store oxygen chemically. Then they have organelles that are specialized. They have myofibrils, which we'll see in a second. Those are contractile proteins. They have a tube organelle, tube network, that's Sarcoplasmic Reticulum. That helps them store and release calcium ions. And then they have structures called T-tubules. T-tubules are part of the sarcolemma. When that sarcolemma or cell membrane goes across the cell, there are places when it dips down and then dips back up again. And that tube that goes inside the cell, that is a T-tubule. So let's start to look at their microscopic anatomy. Myofibrils, remember, are an individual structure made of contractile proteins, and skeletal muscle cells are full of them. It's the arrangement of proteins within the myofibril that gives it the striation. And in fact, the striations are named. The dark part is called the A band. The light part is an I band. If we take one myofibril and magnify it so we can see what's going on, we would see that that myofibril is arranged as repeating functional units called sarcomeres. A sarcomere runs from one Z line, it's a wiggly line there that's made of a structural protein, to the next Z line. So all of this is one sarcomere. The events of contraction follow along one sarcomere, but realized a myofibril can be thousands of sarcomeres long. Within the sarcomere, you can see you have overlapping proteins here. Okay, there's a structural protein that is our Z line. Then we have a blue line that comes out here. That protein is very, very thin, so it got called the thin filament. So you can see the thin filaments are attached to the Z discs and they come and stick out like that, but they are not attached in the middle. Then sandwiched between thin filaments, we have this thick red one. It's kind of fat under the microscope, so it got called the thick filament. Each of these two filaments is made up of proteins, and you can see the names of two of them there, actin and myosin. If we go back and start to look at the individual filaments, in the thick filament, you would see that it's hundreds of molecules called myosin. Myosin has a long tail region and then two heads that stick out. What myosin can do is myosin can bend at its tail and bend at its heads. In fact, remember that the myosin is between the thin filaments. There's a binding site on myosin's head for that thin filament. If you allow it, myosin will bend at its tail, its heads will form a bond with the thin filament called a cross bridge, and then that head will pull in that direction. It'll grab the thin filament and pull on it to move it, and that movement of its head is called a power stroke. And that's what's special about myosin. The thin filament is actually made up of three proteins. The blue one in the background is called actin. Actin has a binding site on it for myosin. Myosin wants to bind right there. It wants to cross bridge right there if you let it. We'll realize if myosin binds actin, it will power stroke, and that's the main event behind a contraction. And there are times when you want a muscle to contract. There are times when you do not want the muscle to contract. If you don't want the muscle to contract, you have to cover up that binding site for myosin. And you have to control whether it's covered up and when it's not covered up. And that's the job of the other two proteins. This long orange one is called tropomyosin. Tropomyosin sits on top of actin, and you can see it covers up the active site. When the active site is covered, myosin cannot bind. That's lovely, but we want to have a way to push tropomyosin off that site whenever we want the muscle to contract. Whenever we want myosin to bind, we must push tropomyosin out of the way. That's the job of troponin. Troponin is sitting here, and troponin knows when to push tropomyosin out of the way to allow myosin to bind to actin. It also knows when to let tropomyosin back so that myosin does not bind actin. Troponin's ability to do that is related to calcium and whether calcium is available in a cell, and calcium's availability is related to an action potential. And how all of these things interact together is the sliding filament theory. Okay. So when these things are overlapped on each other, you can see here's the thick filament made of myosin. Myosin has tails and heads, and each of these heads wants to bind to that actin right there. I'm going to ask you two questions, and if you don't know the answer, then you need to immediately stop, review, and look in your textbook. Question number 1. What's the name of the protein on actin covering the binding site? Question number 2. What is the name of the protein that helps you move that first protein out of the way to allow myosin to bind, or allows that protein back to prevent myosin from binding. Again, if you don't know, you need to stop the slide and do some reviewing. Okay, sarcoplasmic reticulum. Let's see how those myofibrils are sitting in relation to everything else. The myofibrils in a skeletal muscle cell, remember, are packed. So every single one of these structures are a myofibril, and their sarcoplasm is flowing all around them. The myofibrils are kind of surrounded by this blue interlocking network of tubes. That interlocking network of tubes is an organelle. That is sarcoplasmic reticulum. The tubes of the sarcoplasmic reticulum come together and form these blind end sacs. Okay, see, and by blind I mean you can come in, but you can't go out the other end. These blind end sacs are called terminal cisternae. They are kind of the ends of the sarcoplasmic reticulum. Calcium gets stored in the terminal cisternae and in the sarcoplasmic reticulum. And the job of the sarcoplasmic reticulum is to pump calcium from the cytoplasm, store it, and then when an action potential is received, the terminal cisternae will release calcium into the sarcoplasm. Well, right next to the terminal cisternae is the T-tubule, and if you follow the sarcolemma at the bottom here, you can see how it pokes down and makes a tube-like structure. The T-tubule is part of the sarcolemma, so if an action potential comes down the sarcolemma, that action potential will also travel down the T-tubule. When the action potential travels down the T-tubule and lands right here, that action potential in a T-tubule is the trigger to release calcium. And this side, we're just looking at the neuromuscular junction, the fact that we have this synapse to start that whole event, to trigger the action potential on the sarcolemma. You need a neuron to synapse. It's a traditional synapse. So you still have telodendria, you still have axon terminals, you still have synaptic vesicles. An action potential has to come down. On the axon terminal, that opens a calcium voltage gated channel. Calcium flows in. The influx of calcium in the terminal causes exocytosis of neurotransmitter. Neurotransmitter diffuses across the cleft, where it binds to its receptors on the postsynaptic or membrane. Here it's the receptor. Oh, that's the inside. Here's the receptor, the big purple guy. If you can get enough neurotransmitter binding so that you depolarize the sarcolemma to threshold, you will generate an action potential. And it is a synapse, so the same rules apply. You still need enzyme in there to break down neurotransmitter when you don't need it anymore. Alright, so how do muscles contract? So let's put it all together. An action potential comes down the axon, and that axon synapses on a neuron. If you get enough neurotransmitter, you depolarize the sarcolemma to threshold. That generates an action potential on the sarcolemma. The action potential will travel across the sarcolemma and down a T-tubule. When the action potential gets near the T-tubule, or in the T-tubule, that causes calcium to be released from the terminal cisternae. When calcium is released from the terminal cisternae, it flows through the cytoplasm. And it flows across the myofibrils and binds to troponin. When calcium binds troponin, that's the trigger for tropomyosin to change its shape. That allows tropomyosin to move. When tropomyosin moves, the active site is uncovered. Myosin will now form a cross bridge to that active site on actin, and myosin will power stroke. The end result of that is this. Here's my thick filament. Here's my thin filament. The myosin heads, and it's always more than one, form the cross bridge, and they power stroke. And when they do that, this actin molecule gets pulled that way. On the other side of the sarcomere, same event happens. That actin molecule gets pulled that way. What's the end result? The sarcomere shortens. Now, if you want to relax this muscle, now ATP shows up. ATP binds to its little site on myosin's head, and ATP breaks the cross bridge. That's pretty messy, but you know what I mean. So this here, these events going this way, allow a muscle to contract. And notice you needed no ATP for contraction. You need ATP to go the opposite direction. You need ATP to do relaxation. Because ATP, when it shows up, it breaks the cross bridge. Myosin lays down, actin slides back into place, calcium pops off troponin and gets reabsorbed. You get the picture. The best thing to do to see how these things work is to actually use the animations on the interactive physiology disc. Alright, so hopefully you've stopped. Hopefully you've reviewed all of that. Hopefully you understand the sliding filament and the neuromuscular junction. So we're going to talk briefly now, and what's going to come up now is pretty much a lot of terms. Refractory period. The refractory period is the time when the muscle is unable to do another contraction cycle. So similar to being unable to do another action potential, because they are tied. Skeletal muscle tissues refractory period with their contraction cycle is very short. Remember, they're fast contractors. You stimulate them, they contract, they relax, and then they're ready to do it again. So that allows them to a lot of cycles in a given period of time. It is also why they will run out of ATP and energy, and they'll tire easily. Cardiac muscle tissue, other story. Cardiac muscle tissue cells have a very, very long refractory period. That does a couple of things. It makes sure that every single cell in the bundle is back to rest before it contracts again. It helps make sure that they're all contracting at the same time. It also helps ensure that this is a tissue that isn't going to tire easily. So some more terms that we're going to talk about. We're going to look at motor units, a muscle twitch, a graded muscle response, actually several of them, something called TREP, the deal with muscle tone a little bit, different contraction types, and here we go. The motor unit. A motor unit is one motor neuron and all the skeletal muscle cells that it supplies. So you can see this is a spinal nerve in this example. This motor neuron comes out, it branches, and it's attached to cells, muscle fiber cells. Weight-bearing muscles tend to have fewer large motor units. So the muscles in your legs tend to have motor neurons that branch to a lot of skeletal muscle cells. Muscles that accomplish fine movement are the exact opposite. They have a lot of very small motor units. Here's the deal. If you fire off that motor neuron, chances are the cells that it controls are going to contract. A large motor unit means fire an action potential down one motor neuron, you might have 100 cells contracting. If you fire a action potential down a motor neuron of a small motor unit, you could have as few as one cell contracting. This allows you to have tremendous control over the force you generate when you contract skeletal muscles. Now we're going to look at a muscle twitch. A muscle twitch, this is called a myogram, it's a graph. A muscle twitch is a muscle's response to one brief stimulus. And you can see three phases. Excuse me. The latent period is a time when the muscle is not contracting and therefore not generating tension. What's happening here is you have to go action potential, down sarcolemma, down T-tubule, calcium released, calcium binds troponin, that causes tropomyosin to move, myosin makes a cross bridge. All of that's got to take place in the latent period. Once myosin begins to power stroke, that's right there, now the sarcomere shortens and now the muscle's generating tension. And those myosins don't all cross bridge and power stroke at the same time, they do it in turn. So it takes a little bit of time for that muscle to reach a peak generated tension to get the maximum number of cross bridges formed for the stimulus. At that point, once you hit peak, ATP shows up, breaks the cross bridges, and everything starts to slide back down. You lose tension. So you have a latent period, a period of contraction, and a period of relaxation. Different tissues, different skeletal muscles have different types of cells in them, and so they have different twitches. A muscle that's a fast twitch, like your lateral rectus, you can see doesn't take it very much time at all to go contraction, relaxation. Incredibly short time. So this muscle is going to tire out easily. The opposite muscle is a muscle in your calf called the soleus, and you can see this one's actually a slow twitch. Those big weight-bearing muscles tend to be slow twitch fibers because they've got to walk you around and stand you up and carry you throughout the day. So they need to be able to do that nice and slowly so they don't tire out easily. Now the deal with muscles. Skeletal muscles are not all or none. Not all or none, which means they can respond differently to different stimuli. A small stimulus, you get a small contraction, meaning if I need to pick up a pencil, the muscles that are going to generate the tension and the force to move those bones only have to stimulate a few cells to generate a little bit of force. If I go to pick up a grand piano, those same muscles need to generate a lot more tension and force, so they're going to contract many, many more cells. Remember, motor units allow me to control that. You can also change how a muscle responds by something that's very similar to summation. I can make a muscle respond differently by stimulating it more and more frequently, so I can make it contract more times in a given time frame, or I can change the strength of my stimulus. I can give it a stronger stimulus. There are three types of graded muscle responses we're going to look at. One's wave summation, incomplete tetanus, and tetanus. And as I said in class, this tetanus has nothing to do with stepping on a rusty nail. Right, here's the deal. This first one right here is a muscle twitch. Remember, a muscle twitch is one stimulus. So it's a latent period, a period of contraction, a period of relaxation. If I twitch a muscle, if I stimulate it to contract, I allow it to relax a little bit, and then before it has a chance to relax all the way, I stimulate it once more, that's called wave summation. So wave summation is two stimuli. And you can see that the peak, the tension generated on the second twitch, is greater than the first, and that's true. That happens because more calcium is released. Remember, this muscle cell was not allowed to relax 100%. So when you went to do the second stimulus, there was still calcium in the cytoplasm. You stimulated it again, you released even more calcium. That meant even more troponin, even more tropomyosin moved, even more cross bridges formed. So that's wave summation. Wave summation is two stimuli. The second peak has greater force. Incomplete tetanus is multiple stimulations, but you can see the muscles allowed to relax a little bit between each. And what happens in incomplete tetanus is the same concept, even more calcium. So each time you have a greater and greater force. However, all good things must come to an end. If you stimulate a muscle so fast that it no longer has the ability to relax between contractions, it will generate maximum force, stay there, and then force a complete and total relaxation. If in the body you're not able to do the complete and total relaxation, what happens is this muscle will fuse. It will lock in position and stay contracted. Sometimes people have had that experience where they ran for so long or they held an object for so long that the muscle kind of freezes and locks in place, and they have a hard time getting it to relax. You have to massage it out to relax it. That is complete tetanus. So of these three, of the four, remember, a twitch is one stimulus, wave summation is two stimuli. Incomplete tetanus is multiple stimuli, but the muscle's still able to relax a little. Complete tetanus is the stimuli is so fast the muscle cannot relax at all, and it locks in position. You also have TREP. At first glance, TREP looks a little bit like wave summation or incomplete tetanus. But what's different is it is multiple stimuli. You are getting greater and greater tension each time to a maximum peak. However, the muscle is allowed to relax each time completely. So what you're doing is contracting the muscle, allowing it to relax, and then just contracting it right off the bat. TREP occurs, again, because you're increasing calcium in the cytoplasm. Also, when muscles when muscles contract repeatedly, they heat up, they warm up. And that increase in energy makes enzymes more efficient, and reactions tend to happen a little bit faster. So that's TREP. TREP is multiple stimuli, but you allow it to relax fully between each contraction. You get more force. Briefly, muscle tone. We've talked about tone earlier in the semester. Tone is the fact that even if you think a muscle is fully relaxed, it's not. There's always a few cells that contract, so you always have a couple of motor units contracting in turn. They take turns. There are spinal reflexes that allow you to do that. There's somatic, there's autonomic tone. And it's the response to stretch receptors. It keeps the muscle healthy. It keeps it stimulated. All right, contraction types. Turns out you can contract muscles to generate force. That's what we've been talking about. But you can do that in different ways. The traditional way we've just talked about is isotonic. Okay, isotonic means you generate tension by changing length. The opposite to that is isometric. Muscles can also generate tension without changing their length. An example of that is if you hold your arm straight, your arm is straight, your elbow straight, and you still lift a heavy object, the muscles of your hands and your forearm and your arm are generating tension, but that muscle's not contracting. Isotonic, there's two ways you can do that. If it's isotonic concentric, that's the traditional way. Sarcomere shortens to generate tension. There's also this weird, crazy way that's eccentric. And what happens in eccentric, a muscle generates tension, but it actually elongates. Your book gives an example of walking up a steep hill and having your calf muscles have to kind of stretch while generating tension. So muscles can generate tension in these three ways. It's still sliding filament theory. Isometric and eccentric aren't entirely understood as to how the muscle actually does this. All right, muscle metabolism. Muscles have to generate ATP in order to relax. Now, there's a few ways it can go about doing this. One way is to simply recycle its ATP using a molecule called creatine. It's a fast way of just generating or recycling an ATP. Okay, even though it's fast, it doesn't use any oxygen, and the deal is it's very one-to-one. So muscles can run out of that pretty quickly.